laboratory technique used to separate and identify substance in a mixture. In the food industry, chromatography is commonly used to analyze food colorants and additives to ensure the products are safe and meet quality standards. Food colorants are substances added to food to make it more attractive and appealing. Meanwhile, food additives are used to improve taste, texture, freshness, and shelf life. These substances are commonly used in processed foods and beverage.
Now I will explain the four main of type chromatography. Firstly is absorption chromatography, which separate a substance based on how strongly they stick a solid surface. Second is gas chromatography, which is used to separate volatile compound using inert gas. Third is liquid chromatography, where the liquid carry the sample and the compound separate based on their solubility and interaction with the stereotory phase. Lastly is affinity chromatography, separate substance through specific biological interaction and is commonly used to purity protein. Now I will explain about how the paper chromatography works. A small amount of the sample is putted onto photography paper. The paper is placed into the container with a suitable solvent. The solvent rise up the paper by capillary action. The sample dissolves and carry along with the solvent. Different components travel at different rate due to their solubility and affinity for the paper. Components separate into individual spots as they move. The separate spot can be observed and used to identify the substance in the mixture.
Next, there are 10 steps for method and experiment. Step 1. Use a pencil to draw a line approximately 1 until 2 cm from the bottom edge of a sheet of chromatography paper. Step 2. Label each sample as 1, 2, and 3 or any indicator that suits the experiment. Then, place minimum possible amount of the solvent onto the pencil line. Make sure the spot is interval to each other. Step 3. Use a suitable solvent and pour into container beaker to form the shallow layer of the solvent in the beaker. Step 4. Suspend the paper chromatography paper vertically in the beaker. Let the solvent level reach the spots below the line on paper chromatography. Step 5. Cover the container to make sure the atmosphere in it is saturated with solvent vapor to avoid evaporation. Step 6. Let the solvent slowly travel up the paper. Different samples will travel at different rates. Step 7. When the solvent approaches the top of the paper, remove the chromatography paper immediately. Step 8. Use a suitable solvent and pour into container to form the shallow layer of the solvent in the beaker. Step 9. Mark the solvent front with a pencil. Allow it to dry. And then lastly, step 10. Observe the separated spots and record their positions. Distance traveled by solvent and sample component is measured to calculate the RF values.
In the food industry, chromatography helps manufacturers identify food colorants and additives used in products. It is also important for detecting harmful substances and ensuring only approved additives are present in food products. For example, chromatography can be used to test the food coloring in candies, detect preservative in soft drinks, and ensure that only approved additives are used in food products.
For the example results, we're taking candy chromatography for our example. In this experiment, different candy coatings were tested using the paper chromatography method. Like you can see, in the right side of the slide. So for the results, the red candy remains red in color. Orange candy has red and yellow spots. Purple candy has blue and red spots. And finally, green candy has blue and yellow spots.
Now I will explain advantage and limitation of chromatography. The advantage is simple and easy to perform. Separately different food colorant effectively require only small sample amount, fast and accurate analysis, help identify harmful or annotation additive, also cost effective and for laboratory testing. For limitation, some method require expensive equipment, need skill handling and interpretation. Certain compound may not separate completely. Example, preparation can be time consuming. Result may be affected by contamination. Some technique use hazardous chemical or solvent.
In conclusion, chromatography is a useful and reliable technique used for separating and identifying food colorants and additives. The technique demonstrates how different dyes can be separated based on their solubility and attraction to the chromatography paper. Therefore, chromatography is important in the food industry for quality control and food safety.